Now look at this.
What's up, Poopy Bio Penguins? Today we do topic 6.8 on biotechnology. So first up, let's talk about gel electrophoresis. Gel electrophoresis is going to be used to separate either proteins or DNA by size or charge. So the DNA, in this case, is going to be inserted into the well. So once it's inserted in the well, it's inserted at that negative end. We turn the electricity on, and it's going to get drawn or pulled toward the positive end. When it does this, it's going to separate those DNA fragments by size. Smaller fragments can go a far longer distance, and longer fragments go a shorter distance. The reason is that there's little pores inside the gel, and so smaller fragments are going to be able to weave in and out of those pores faster than a larger fragment can. The reason why there's multiple fragments is due to the fact that we have restriction enzymes. Restriction enzymes will look for specific sites called restriction sites to cut. So if we have an individual that's homozygous dominant and an individual that's homozygous recessive, they're going to have different restriction sites, which is going to cause the DNA to be cut differently. So we can compare these two individuals. So this has been used on the AP exam in the past in combination with a pedigree, in which you could determine individuals' genotypes based on the gel or based on the pedigree, or you'll combine the information between the two. So PCR is going to stand for polymerase chain reaction. This is set up using a thermocycler. So what's going to happen in the thermocycler is that it's going to heat up, and it heats up to a very high temperature. That high temperature is going to denature or pull apart the two strands of DNA. There's going to be a cooling down process. That's when a primer is going to bind. So the primer is going to be looking for a certain gene of interest that we're making copies of. And then the last step, DNA polymerase is going to then replicate that segment. So we can use PCR to make multiple copies of a segment of DNA in a very short amount of time. PCR uses a very specific type of polymerase. It's going to use a heat-resistant polymerase that's coming from thermophiles. Thermophiles are individuals that live in a very high temperature environment, and so we're going to use Taq polymerase traditionally in this experiment. So you all probably did a transformation experiment. In the transformation experiment, we're going to heat shot bacteria in order to transform or add a plasmid from the environment into the bacteria. We then are able to test the bacteria whether it was transformed or not by growing it on different plates. So I searched AP Bio transformation experiment on Google and found this beautiful image. Due to the transformation, these ones were not transformed and these ones were transformed. So we're going to be able to determine that the experiment was viable because of the fact that they both got lawns on the ampicillin plate, but only those transformed bacteria were able to grow in the presence of ampicillin because the plasmid provided them the resistance gene to be able to grow in the presence. In DNA sequencing, you use radioactive nucleotides, and then you use PCR to be able to determine which nucleotide was in which position so you can find the DNA sequence.